Science friends. Today we are talking about cell division, or in other terms, the cell cycle. And so the cell cycle discusses the life cycle of a cell. So basically, how do cells reproduce? So cells constantly have to reproduce because we're always sloughing off old cells, getting rid of cells, and creating new cells. So this is particularly important as organisms are growing. So from the time you were a child, you had much fewer cells to what you have now. And so that growth is going to necessitate a lot of cell division. But even just for adults, cells have finite lifespans. For example, a red blood cell lives about 90 days. And then after that, you're going to have those red blood cells replaced. And so cell division is what does this process. So in the process of cell division, we take one cell that's known as our parent cell, and we divide it and split it in half to form two daughter cells. Okay, now that's super important, these parent cells and the daughter cells. So cell division happens in somatic cells. And what somatic cells are, the word soma means body. Okay, so these are body cells. They are non-sex cells. Okay, all the cells in your body, with the exception of sperm or egg, are somatic cells. And these somatic cells are produced by this cell cycle process called mitosis. Okay, these two daughter cells that are produced are genetically identical. In humans, they have 46 chromosomes, okay? That's 23 pairs of chromosomes. One of those pairs you inherited from your mom, and one of those pairs you inherited from your dad, okay? And so that makes up our 46 chromosomes. Now that number is different for every different type of organism, okay? And we'll talk about that more a little bit later on. Okay, so that's somatic cells. Now, the other type of cells, the sex cells, are called gametes. Now, they are produced by a different process, and that's called meiosis. We will talk about meiosis more in our genetics unit. Okay, these cells that are produced in meiosis are genetically unique. They are different. Okay, so one sperm cell and another sperm cell are very different. One egg cell and another egg cell are very different. That's why you can have fraternal twins, right? Two babies that were born at the same time, but they were fertilized with different sperm, or had different eggs. And so that makes them have different DNA, and they are genetically unique. Unlike our body cells, our somatic cells, which are all the same. All of your red blood cells are identical to one another. All of your skin cells are identical to one another genetically, okay? So gametes, these sex cells in humans, have 23 chromosomes. And that's important because 23 in the sperm is going to combine with 23 in the egg to make 46. Okay? So, while we're not going to get into the process of meiosis today, I want you to see the similarities and differences between mitosis and meiosis. So, number one, mitosis is going to produce genetically identical cells. Okay? It's going to make two of them. Meiosis is going to produce genetically unique cells, and it's going to make four of them. Okay? Mitosis is going to produce cells that have the full chromosome number. Okay? That's referred to as the diploid number of chromosomes, which in humans is 46. Meiosis is going to produce half of the chromosome number, which is 23. That's referred to as the haploid number in humans. Okay. Mitosis is going to take place in those somatic cells. Meiosis is going to occur in the gametes. Okay. So we are going to focus on mitosis in this unit and talk all about mitosis and what happens. The processes are very similar, and we'll come back in genetics and learn more about meiosis. Okay, so this is a cell right here that appears to be undergoing cell division. Okay, so here's our cell membrane. This is our nucleus. And the DNA right now in this form where it just kind of looks like squiggles, that's called chromatin. Okay? When the cell is not dividing, DNA exists as chromatin. Okay? As the cell starts to divide, in the first phase of cell division, the DNA condenses into chromosomes. And you all probably know what chromosomes are. They look, something like that. Okay, like this. Condenses into chromosomes. But you can only see chromosomes when the cell is actively dividing. Okay, when the cell is just in an inactive, I shouldn't say inactive, but a non-dividing state, you don't see chromosomes. DNA exists as chromatin. Okay. And then it condenses. And when I say condenses, what I mean is, and we'll talk about this more later, is it winds up, okay? So the DNA winds up to form these chromosome structures, okay? It coils, it condenses to form the chromosomes, okay? So, in cell division, in mitosis, we have what are called sister chromatids, and that's the copy of the original chromosome, okay? So when the DNA is replicated, you have this one chromosome, and it gets copied, and you form the sister chromatids. And these sister chromatids then separate into two individual cells, okay? This is mitosis, okay? What do you notice about this cell and this cell, or this cell and this cell? You should say they are genetically identical. Those chromosomes look exactly alike. The DNA molecules, if you compared the bases, the A's, the T's, the G's, and the C's, they would be identical. Down here, we made two cells, which is what I told you we would make in mitosis. So we took this cell, this first one, called our parent cell, and we made two daughter cells, okay? And those sister chromatids are the copies, joined at an area in the middle called the centromere. So you can kind of see the centromere here. You can kind of see this pinched area here. That's referred to as the centromere. Okay? So, the cell cycle includes a lot of phases. The biggest part of the cell cycle is interphase. Interphase is broken down into three other phases: G1, S, and G2. You gotta know this. You gotta know this, okay? G stands for growth. So the cell's gonna grow and, prepare for division, okay? S stands for synthesis. That is when the DNA is going to be newly synthesized. That's when the DNA is going to be copied, okay? Because in order for these chromosomes to divide, they have to first copy themselves, okay? And so that happens in the S phase, all right? And then G2 is a second growth phase. So the cell continues to grow, okay? A very small part of the cell cycle is mitosis, okay? So sometimes people will mistakenly exchange the terms mitosis and cell cycle, and that's not exactly correct, okay? Mitosis is just a small part of the cell cycle. Some people will refer to mitosis as cell division, and that is technically correct because the mitosis is where the division of the chromosomes happens, okay? All right, so we've got interphase, mitosis, and then cytokinesis, okay? That is the main phases. Now, within mitosis, there are 4 main phases. In AP, we kind of add this 5th phase here, prometaphase. But what you may find helpful is IPMAT-C. IPMAT-C. That's an acronym to help you remember the phases of the cell cycle. So I stands for interphase. That's this first phase here that takes the longest part. The interphase includes G1, S, and G2. The PMAT is what really makes up mitosis, which sometimes you will see it referenced as the M phase. Not often. Usually it's just referred to as mitosis, okay? But PMAT is mitosis. That stands for prophase, metaphase, anaphase, and telophase, okay? And then the C is the last part, which is cytokinesis, okay? And at the end of cytokinesis, that's when you have your 2 new cells that have formed. So let's walk through these phases together. You have to know what happens in each phase. This is basic biology. We teach this in the generalist of general biologies. You have to be able to know these phases, okay? So interphase, okay? In interphase, this is G2. So that means it's already done G1 and S. The DNA has already been synthesized. It's already been copied. So here we have this chromatin. Remember, it's not coiled up into chromosomes yet. But because it's already past that S synthesis phase, it's already been duplicated. Okay? The cell membrane, the nuclear membrane, you can see these little slits here, is starting to break down. Okay? That is interphase. In prophase, you see these chromosomes starting to condense. It doesn't look like chromatin anymore. The chromosomes are starting to condense, and these spindle fibers are starting to form. Now, if you remember, we have very briefly one time talked about spindle fibers. Does anybody remember when? We talked about spindle fibers when we talked about motor proteins, okay? Spindle fibers are made, have these microtubules. You see that term there. You should recognize microtubules. So the spindle fibers form these microtubules, and that's what pulls the chromosomes apart. They use those motor proteins that walk on those microtubule train tracks to move the chromosomes around within the cell. Okay, that's kind of an aside. Back here to prophase. So in prophase, we see the chromosomes forming. Okay, the centrosomes are starting to move to the poles. They're going to move to the end of the cell. By prometaphase, they're all the way at the end of the cell. These microtubules are fully kind of formed there. The chromosomes are more condensed, okay? And the microtubules are connecting to the centrosome of the centromere of the chromosome, okay? So I don't care that you really know prometaphase quite as much. It's not. It's just in the middle, okay? Metaphase. Metaphase is really important. Think metaphase, middle. All the chromosomes line up on the middle, okay? And the microtubules are attached to the centromere of the chromosome, okay? In anaphase, these sister chromosomes are starting to pull apart. The sister chromatids are starting to pull apart. And then telophase, you see the nucleus starting to reform. The chromosomes are starting to decondense. The microfilaments are gone, and they've all kind of coiled back here in the spindle fiber. And then you see this area right here where the cytoplasm is starting to pinch together, okay? That's called a cleavage furrow. You need to know that term, a cleavage furrow. And that is going to be the start of cytokinesis. And then in cytokinesis, this would fully divide, and you would have two separate cells, okay? So here again, you see the cleavage furrow right here. That's going to separate those, and then this is the daughter cells, the two full daughter cells. Now, important to know that the cleavage furrow is only in animal cells. In plant cells, you don't have a cleavage furrow, but you have something that's called a cell plate that forms and divides the two daughter cells of a plant cell, okay? And that's a little bit different because of the cell wall, all right? Here is that diagram, like I was kind of talking about, where these motor proteins attach onto a microtubule and allow the chromosomes to move on the microtubule, okay? And we talked about that a little bit when we talked about motor proteins and microtubules and why they were important in the cytoskeleton, okay? Now, binary fission is kind of a different type of cell division that happens in bacterial cells, so prokaryotes, and then some single-celled eukaryotes. And so it happens a little bit differently, but it's basically mitosis, okay? So this is how bacteria replicate. Now, I just told you that individual cells replicate in mitosis, right? But not sex cells, skin cells, hair cells, heart cells, lung cells, gut cells. All the other cells replicate with mitosis. But binary fission is the way that this whole organism replicates, okay? But just in these bacteria. And so it's kind of a modified mitosis that allows bacteria to replicate the whole organism. Not just a cell, like in mitosis, the whole living thing, okay? So, if you have any questions about that, let me know. You have a sequencing assignment today. You should have a worksheet in your binder about sequencing. And so I want you to go through and put those in order, and then see if you can label them as to what phase they are too. There's obviously more than like IPMAT-C on there. There's, so there's going to be like early prophase, late prophase, early metaphase, late metaphase. So I'm not looking for those names specifically. I'm going to be grading for the numbers, but I think that's a good practice to kind of help you be able to identify them and where those stages fall in the process. So let me know if you have any questions, and you'll have a great day.